My research involves genomic dark matter, and we try to understand how genomic dark matter regulates our generation of diseases and pathogenesis of different complex phenotypes and complex diseases. We finished one last project where we found new element inside the dark matter of genome, and we found that this element is highly expressed, and when it's expressed and in your genome, then it helps to protect brain against the Parkinson's disease and helps your neurons to survive. My name is Sulev Koks, and I work at Murdoch University and at Perron Institute. I'm professor of genomics, and genomic dark matter is matter in our genome. It's actually part of our genome that we don't understand at all. And the good thing for us is it's actually 98% of our genome we don't understand. We only understand genome like 2% of it. And the rest of it, we just don't understand what it is doing, why it's there, why do we need it. But we know that we need it because it's some of this genomic dark matter, or most of it, is actually human specific. And we know that there are parts of this dark matter that actually makes us human, that actually help us to make us to talk, make us to walk, make us to lose tail, for instance. We don't have tail. Mammals have tail. Most of mammals have tail. We don't. And this is why we don't have tail, is actually in this dark matter. We actually have the gene. We have the gene. We can have a tail, but thanks to dark matter, we don't have it. So we don't understand how does it work. And while the tail sounds like it's a bit like vanity or cosmetic thing, but it's biology, and understanding these kind of principles can help us to understand also diseases. And the paper we published with the help of Pawsey is about one element they found in our genome. We call them elements. So it's genomic dark matter. They are not even genes. Because they even don't look very much like genes, but they function like genes. They are like some bits of it. Let's put it this way, but we call them elements. And one element we found is actually neuroprotective. It helps us to, it protects us, our brain, against neurodegeneration. And we found that Parkinson patients or people, we age, our risk to get Parkinson disease will increase while we age, but we know that we start to lose some neurons there in the brain. But then we found that some patients actually lose it very fast. They are actually deteriorate very quickly, and they need more drugs, and they really start to get dementia very quickly. And then we found that, these patients don't have this element. And the patients who have this element in the brain, they actually are doing very fine. And we found this element, and the paper is very much about how did we find it. And then also we try to understand the function, what it does in our genome, and how it actually protects our neurons from dying or just how it's actually neuroprotective. And and we used Pawsey supercomputer to do most of it, some of the work, even most of this work, because it was we had the population we work with is actually a very large cohort of Parkinson patients, and they have been followed for 5 years. We have annual follow-up data, and we have whole genome data. So basically, to analyze these elements we work with, we use whole genome data only, and per patient or per subject, we have usually the data volume is almost like 100 gigabytes. So we need like supercomputers to work this data, this massive amount of data. It's really massive. Yes, the key thing is that we have actually proved that in our dark matter we can have elements that are actually useful for our genome, and this is why we have it. And people always thought that it's junk, but we proved that now it's actually can be useful also. We need everything in our genome. We need dark matter and we need other genes there. What this element does in our genome, it's expressed, and when something is expressed in our genome, we see RNA. We have DNA and RNA. And RNA is always RNA always shows you that something is active in our genome. And these elements can be also active. many of them actually are quite active. And this element we found is actually transcribed, and we have RNA. And what we do right now, and partially we showed also in this paper that it's potential that if you can transfer, we can use this RNA as a drug. We can actually make a drug for those people, or even early Parkinson patients. We can give it as a drug to help to preserve the neurons or the cells in the brain they still have there, and just to delay the Parkinson. So we can actually develop RNA-based drug for Parkinson's disease. And there already are some drugs, not for Parkinson, but other rare neurological conditions where we actually transfer a new gene by replacing it. We just replace a broken gene, or we give another copy of so-called good genes that actually can help to preserve cells inside the brain. And this technology is actually there, and it's possible to do it. And especially here in WA, because we have also experience and competence here. We have people who actually have done it before here in Perth. Our research is based on actually very large international collaboration. We work with US scientists in the US, Canada, in Europe, in Asia, in Africa, South America. So it's really like we have a very large cohort of patients that are actually collected from different parts of the world. First, we get the clinical data, then we collect biosamples from patients, and biosamples are blood samples, even urine samples, and from blood we collect like RNA, DNA for genotyping, and RNA to see what parts of DNA are activated. And then the labs in different places of the world, they do the analysis of these samples, and then we put all our information together. All the data will be shared, and it's really very much like so-called open science, or whatever you call it. And it's a good example of how scientists can work together. And as we had very good clinical information and very good genomic information, we could start to work. But this information was massive. It's massive. Really, the data we have is, I think it's 200 terabytes altogether, even 250 terabytes at least. It's basically raw data, our primary data we work with. So already copying it and transporting it all over the world will take time. And this is where I contacted Pawsey. It was a massive asset. And I would say that out of all this collaboration network I work with on Parkinson, our center is actually the best equipped with computational options here, because really Pawsey is really very powerful. We couldn't have done this research without Pawsey. Absolutely, there's no question. We have like 2,000 patients. And then we genotype all of them. We try to, in every patient, we can try to draw a map how many those elements they have. We call them transposable elements. These are dark matter elements, are transposable elements, because they can jump around. They can actually, in your lifetime, they can jump around in your genome, and they can cause you a mutation, like you acquired it during lifetime. This is the name. And every patient had them, maybe 10,000 of them in every genome. So it's basically for every patient we have this file where we have positions and the type and all the information describing these 10,000 elements that we study. And we had like 2,000 patients. So this is already quite a large amount of data. And then we start to compare. We compare, of course, because we have also half of the population, or the study group is actually controls. So it's a healthy age match, sex match, healthy controls. And of course we compare the frequency and the type what we have there. Are some of them actually causing Parkinson, or do we see they are more frequent or less frequent in Parkinson patients? But as we have also longitudinal data, then we can start to model how this genomic or genetic information models our clinical outcomes. This is because we have imaging data. We have MRI imaging data, or PET scans, or PET scan, where we have measured the degeneration in the brain, how many dopaminergic neurons they have in the brain left in patients, and we see the decline quite. In some patients it's very quick, some patients it's very stable, but we see that it's much less than in healthy controls. What we do on Pawsey, we just combine and analyze. First of all, we call the genotypes, and then we do some kind of modeling there as well. We use machine learning in our process, where we need to actually call those elements, those transposable elements, from raw genomic data, because the files are so large, and it's very hard to just to ensure that what we do is actually correct. So we need to we use machine learning basically as a reference, like a virtual reference. One thing is that we get more students, because we already, when we started with this dark matter research, we didn't have very nice and good chunks that we could actually give to students, and we even didn't know do we find anything from there or not. But now we already have something on our table, and we can give it to students, some smaller projects. And we work with the same elements that we published, to make it like druggable, to make it really as a drug and transferable, so we can actually start to transfect cells, and we can actually do animal experiments. This is just one path to develop a drug based on this finding. But we have also other elements where some of them have similar effect, so they basically help you to reduce the burden of the Parkinson's disease, and we continue we try to find some more elements that are potentially druggable, and if they're not even druggable, then just to get more information, understanding how they actually regulate the cells in the brain and what they actually do in the brain.